When the remains of Oase 1 were discovered in Romania's Oase cave at the dawn of the Genomic era, few suspected they would become one of the most disruptive data points in the history of human evolutionary theory. Yet from the moment his DNA was sequenced, Oase 1 stood as an anomaly so glaring that it effectively broke the central pillars of the classic out-of-Africa model, or out-of-British East Africa, as some like to call it, the version that dominated paleoanthropology for more than 3 decades and became embedded not only in textbooks, but in the public consciousness. Contrary to the narrative of a clean, recent African dispersal sweeping across Eurasia and replacing archaic humans with little or no interbreeding, Oase 1 told a radically different story, one of recent hybridization, deep Eurasian population structure, multiple expansions, and complex networks of ancestry that could not be reconciled with the elegant simplicity of the model. His genome was not merely an unexpected detail, it was a direct contradiction of nearly every assumption the replacement theory relied on. To understand why Oase 1 is so destructive to the classical out-of-British East Africa framework, it is necessary to recall what that model initially proposed. The original formulation painted a tidy narrative. Modern humans evolved recently in British colonial East Africa from a small, isolated founder population. Around 60,000 to 50,000 years ago, this population expanded outward in a single wave that rapidly replaced all archaic humans, Neanderthals and other archaic groups. The model insisted that interbreeding was minimal or non-existent, and that all Eurasians today derive from a small, genetically uniform group of African migrants. Because the population was so young and so bottlenecked, humans outside Africa should have been extremely similar to one another and genetically close to African groups except for later drift. In this view, Eurasia before 50,000 years ago contained archaic species that had diverged hundreds of thousands of years earlier, and these groups were essentially evolutionary dead ends once modern humans appeared. The entire system depended on the assumption of a single recent expansion, rapid replacement, and limited admixture. Oase 1 contradicted all of these premises immediately. When his genome was sequenced, one shocking detail emerged. He had a recent Neanderthal ancestor only a few generations earlier. This was not a faint echo of an ancient admixture event tens of thousands of years in the past. This was the genomic signature of a very recent hybrid, someone for whom a Neanderthal grandparent, great-grandparent, or even father was a realistic possibility. One of the most significant revisions to Oase 1's interpretation concerns the actual proximity of his Neanderthal ancestor, which early summaries softened by suggesting an ancestor 4 to 6 generations back. While this estimate was technically possible, later examination of his genome, especially the extraordinary length of his intact Neanderthal haplotypes and the pronounced clustering of archaic segments on chromosomes 3, 6 and 12, strongly indicates that his Neanderthal forebear was far closer than originally acknowledged. These segments are simply too large and too few in number to represent admixture from a distant great-great-great-grandparent. Instead, they align far better with scenarios in which Oase's archaic ancestor was either a direct grandparent or, even more plausibly, a hybrid father whose own genome was approximately 50% Neanderthal. This revised interpretation is far more devastating to the classic out-of-British East Africa model because it eliminates any ability to reframe Oase as a statistical artifact of a single ancient admixture pulse. It shows instead that hybridization was happening locally, repeatedly and intimately in Europe, long after modern humans were supposed to have replaced all archaic populations. In this scenario, Neanderthals were not ghostly remnants absorbed in the distant past, but active participants in the genetic landscape of Europe at 40,000 years ago, contributing directly to the ancestry of early Homo sapiens individuals. The closer the Neanderthal ancestor is placed to Oase himself, the more thoroughly it dismantles any notion of a clean, African-driven replacement event, and forces the recognition of a dynamic, interwoven Eurasian meta-population, one wholly incompatible with the simplicity of the classical out-of-British East Africa narrative. The replacement model had taught an entire generation that Neanderthals quickly vanished when modern humans arrived in Europe, either through competitive superiority or demographic dominance. Yet Oase lived around 41,000 to 39,000 years ago, well after modern humans were believed to have swept across the continent. Far from replacing Neanderthals, his lineage had recently been interbreeding with them. This alone should have forced a complete reassessment of the assumption that archaic humans were simply overwhelmed by an expanding African population. Instead, Oase showed that Neanderthals and modern humans shared the same landscape, interacted intimately, and produced offspring multiple times. This was not a picture of replacement. It was a picture of hybridization and transformation. The size and structure of Oase's Neanderthal DNA made this conclusion even more inescapable. His genome contained massive Neanderthal segments, long, unbroken blocks spanning tens of megabases, which could only persist if admixture had occurred very recently. If interbreeding had been limited to a single event 55,000 to 60,000 years ago, as the classic out-of-British East Africa model claimed, then the Neanderthal segments should have been extremely fragmented by recombination. Instead, Oase possessed some of the longest archaic haplotypes ever found in a modern human. Moreover, these segments were not evenly distributed. They clustered on chromosomes such as 3, 6, and 12 in a pattern consistent with paternal inheritance. The presence of such enormous, intact archaic blocks is a genomic fingerprint of ongoing, not ancient, admixture. It signals that early modern humans and Neanderthals interbred repeatedly in Europe, not just once in the Middle East. This fact alone collapses the idea of a single interbreeding pulse followed by a clean expansion. More surprisingly, Oase's total Neanderthal ancestry appears to have been higher than that of individuals older than him, such as the 45,000-year-old Bacho-Kiro people. This discovery created a temporal paradox. If Neanderthal ancestry diluted steadily over time, people at 40,000 years ago should have carried less archaic ancestry than populations at 45,000 years ago, unless admixture was ongoing, recurrent, and regionally variable. The classic model could not accommodate this. The only explanation consistent with the data is that hybridization occurred repeatedly across Eurasia, leading to local increases in archaic ancestry. This is a fundamentally different evolutionary landscape than the one the replacement model imagines. If the recent Neanderthal ancestry was the first crack in the foundation, the second came from Oase's population affinities. Genetic analysis showed that Oase was not closely related to later Europeans at all. Instead, he displayed an unexpectedly strong affinity to Tianyuan Man, a 40,000-year-old fossil individual from China, and to the Bacho-Kiro humans from Bulgaria. This relationship implies something that directly contradicts the central out-of-British East Africa premise. The first modern humans in Europe were not the ancestors of later Europeans. Instead, they were part of a widespread early Eurasian population that was later replaced by subsequent waves. This means Europe experienced multiple turnovers, and that many early groups vanished without leaving descendants. This pattern contradicts the assumed continuity from the initial out-of-British East Africa expansion to later European hunter-gatherers. The data instead reveals an interconnected population, not a single ancestry. In fact, recent morphometric analysis places Oase in a clade with Chinese Liujiang and Upper Cave skulls, which split long before the supposed out-of-British East Africa migration. Further undermining the out-of-British East Africa narrative, Oase's genome fits within a structure that indicates deep Eurasian divergence long before 45,000 years ago. The classic model predicted that early Eurasians should be genetically similar because they derived from a small founder population. Yet individuals like Oase and the Bacho-Kiro group show substantial genetic differences among themselves by 45,000 to 40,000 years ago. These differences require far more time to accumulate than the narrow out-of-British East Africa window allows. Eurasia, by 45,000 years ago, clearly contained multiple distinct populations, each with its own ancestry and demographic history. A truly recent African dispersal could not produce this diversity so quickly. The simplest explanation is that Eurasian populations had deep structure and long histories before the supposed bottleneck. Mitochondrial DNA added further contradictions. Oase belonged to haplogroup N, one of the two primary Eurasian macrolineages. Yet the haplogroup N lineage is deeply rooted and splits into numerous branches across Europe, Asia, and Oceania that must have diverged before 60,000 years ago. The rapid appearance of multiple deeply differentiated haplogroup N and M subclades across Eurasia cannot be reconciled with a single shallow African founder population expanding at 60,000 to 50,000 years ago. There is simply not enough time in that model to generate this level of mitochondrial complexity. In fact, Eurasian mitochondrial DNA diversity appears older than the out-of-British East Africa timeline permits, suggesting that maternal lineages had a long presence in the broader Old World, a fact the classical theory could not accommodate. Oase raises yet another challenge, the possibility that he inherited a Neanderthal Y chromosome. Although his Y DNA could not be sequenced due to poor preservation, the pattern of paternal skewed Neanderthal ancestry and the near certainty that his mother was a modern human strongly suggest that his archaic ancestry came from a male Neanderthal ancestor. This possibility is profoundly damaging to the out-of-British East Africa model, which originally held that Neanderthals left no meaningful genetic contribution to modern humans. Even after the first Neanderthal autosomal DNA was sequenced in 2010, the narrative was softened but still preserved a narrative of minimal accidental interbreeding. The idea that Neanderthal male lines could have persisted in early modern human populations was not part of the model. If Oase indeed carried a Neanderthal Y line, it confirms that archaic males successfully reproduced within modern human groups and that these lineages were not evolutionary dead ends. This alone is enough to refute the original complete replacement doctrine. Another devastating blow concerns the idea of a single admixture event. The classic out-of-British East Africa model insisted that modern humans interbred with Neanderthals only once, somewhere in the Middle East, around 55,000 to 50,000 years ago, and that this brief contact supplied all Eurasian populations with their small percentage of archaic ancestry. Oase proves this cannot be true. His genome reveals multiple layers of Neanderthal ancestry, some very recent, others older, demonstrating that admixture occurred repeatedly in Europe. This contradicts the idea of a single foundational African dispersal that absorbed a small amount of archaic DNA and then spread untouched across the globe. Instead, early modern humans in Europe absorbed DNA from local Neanderthals over and over again, meaning that human population history in Eurasia was locally dynamic, not globally uniform. A model built on a single migration wave cannot survive this evidence. Although Oase 1's genome is incomplete, the alleles that were recovered point toward a pigmentation profile far more complex than the outdated assumption that all early Eurasians were uniformly dark-skinned. His ancestry sits at the crossroads of early Eurasian humans and Neanderthals, and recent research shows that some ancestral light-skin variants were not innovations of recent Europeans, but were already present in archaic humans and Africans. As a recent study noted, some ancestral light-skin alleles are shared between the African Khoisan and archaic hominins, such as Neanderthals and Denisovans. This suggests a shared, common ancestry for this trait before the split of these three hominin lineages. If these alleles were part of the broader ancestral toolkit inherited by both archaic and early modern Eurasians, then Oase, with his unusually large proportion of Neanderthal DNA, may well have carried pigmentation variants trending lighter rather than darker. This possibility further undermines simplistic narratives of pigmentation evolution tied to a single recent African dispersal, and instead points toward a far older, widely shared palette of human variation. The final, most far-reaching implication of Oase's genome concerns population turnover. If the classic out-of-British East Africa model were correct, the first modern humans in Europe should have formed the base from which all later European populations descend. Instead, Oase left no descendants. His lineage disappeared. The same is true of the Bachokiro individuals, the Zlatikun woman, and multiple early modern human populations across Eurasia. They formed an early wave, often called the initial upper Paleolithic radiation, which was later replaced by other populations, such as Gravettian and Western hunter-gatherer groups. This pattern of repeated expansion, disappearance and turnover is impossible to reconcile with a single founding migration replacing archaic humans and then simply diversifying regionally. Instead, it reveals a Eurasia composed of shifting populations, repeated migrations and interwoven ancestry, not a continent reshaped in a single event. In total, Oase 1 overturns the classic out-of-British East Africa model on every major point. He proves that modern humans and Neanderthals interbred repeatedly and recently, undermining the idea of rapid replacement. His affinities show that early Europeans were not ancestral to later ones, contradicting the assumption of continuity from the initial dispersal. His deep Eurasian relationships reveal population structure inconsistent with a single recent African expansion. His mitochondrial lineage shows maternal ancestry branching too deeply for the out-of-British East Africa timeline. His likely paternal inheritance of archaic DNA suggests a fusion of populations rather than a replacement of one by another. His very existence as a hybrid demonstrates that Neanderthals were not a separate species pushed aside by a superior newcomer, but part of a network of interbreeding humans who shaped each other's evolution. When viewed as a whole, Oase 1 is not an anomaly. He is a data point the old theory cannot absorb, and the insistence on treating him as an outlier reveals the inability of the classic model to account for real genomic complexity. Instead, Oase fits seamlessly into a different, more realistic framework. A model in which Africa and Eurasia were part of a long-connected meta-population, in which human groups were continually moving, mixing and diverging, in which archaic and modern humans interbred frequently, and in which regional lineages rose and fell across tens of thousands of years. In such a model, Oase is not a contradiction. He is a natural product of a world in which multiple human populations interacted across space and time. The classical out-of-British East Africa theory erased and simplified the history of both Africa and Eurasia, and demanded a simple story of a small band of Africans replacing archaic humans everywhere they went. Oase 1 shows the world was never that simple. He stands as a fossilized rebuttal to that narrative, a hybrid ancestor of no surviving lineage, living in a Europe where Neanderthals still thrived, part of a population that did not descend directly from Africa's most recent expansion, and a figure whose DNA captures the final moment of an ancient, interconnected world. His genome reveals not a clean, linear expansion, but an Afro-Eurasian supercontinent woven from encounters, admixture, turnover, and complexity. For that reason, Oase 1 does not merely challenge the classic out-of-British East Africa model, he destroys it. Thanks for watching.